Today, I'm going to be presenting my dissertation, Investigations into Effects of Environmental Stressors on Lake Sturgeon Physiology, Behavior, and Survival during Early Ontogeny. So we're all familiar with stress, and we're all familiar with the idea that stress can change your behavior and your physiology. And we usually think of that as a negative thing. But as Robert Greene Ingersoll once said, in nature, there are neither rewards nor punishments, there are consequences. In other words, to understand whether stress outcomes are positive or negative, we need to be able to define their adaptive value within the environment in which they exist. So if we want to evaluate the role of stress, we have to examine it within relevant ecological contexts. So I'm going to give some background about how stress can be quantified. This is the HPA or HPI stress axis, and this regulates how an individual responds to a stressor. The HPI stands for hypothalamic, pituitary, adrenal, or interrenal, depending on the species. So in response to a threat being perceived, there's this cascade of events that's triggered, and the end result is that cortisol is produced. So cortisol does a lot of important things in the body. It's involved in mobilizing energy, suppressing growth, it's involved in the immune system, and these are all to help the individual cope with whatever the source of stress was. So there are two kinds of stress. Chronic stress is constant or repeated, so this is something that's experienced over a long term. And this can actually cause changes to the way that the HPI axis functions. And I'm not going to go into the molecular mechanism of that for the sake of time today, but it has to do with epigenetic regulation of the receptors that control the negative feedback loop. And if there are changes to the negative feedback loop, that's responsible for shutting the system off after it's been turned on in response to the stressor. So changes to that mean changes in how much cortisol is produced. An example of chronic stress would be climate change. This is something that wildlife populations are experiencing as a stressor over a long period of time. An acute stressor just refers to a one-time specific experience of a stressor. And the way that an individual reacts to that can reveal how they've been affected by any chronic stress that they've been experiencing. An example of an acute stressor would be encountering a predator. So stress can have profound behavioral changes, and this has been very well studied in a variety of model systems. And the behaviors that we see are fairly consistent across species. They all look kind of like anxiety-like behaviors. So for example, in rodents, this can look like reduced risk-taking. In sticklebacks, this involves changes to personality, which is quantified by boldness and aggression. In zebra finches, there's inhibited reproductive behavior, and that involves vocalizations and courtship. So all of these physiological and behavioral changes can have a positive or a negative impact on fitness, depending on whether they're suited or not suited to the environment. So for example, with the zebra finches, the chronic stress that they were experiencing was lack of food. And so limiting reproduction during that time of scarcity is probably a good idea because individuals need to conserve their energy and their resources. So that would be an adaptive behavioral response to that specific environment. And this points to the importance of the stress axis as being a means of plasticity by which individuals can respond to changing environmental conditions. So a lot of these behavioral changes resulting from stress have to do with anti-predator behavior, which makes a lot of sense. If predation risk is the source of stress, then individuals need to be able to behaviorally respond to that in order to potentially avoid predators. So I studied stress using lake sturgeon as a model system. Lake sturgeon are an ancient chondrostean fish species, and they're regionally threatened in the Great Lakes. Their populations are extremely vulnerable to environmental stressors, especially those related to habitat disturbance and climate change. They also have 99% mortality during the early life stages, and this is partly due to the high predation rates that they encounter during the larval stage. So to give you a little bit of background about the lake sturgeon life cycle, lake sturgeon live in lakes, and then every spring they swim upriver for the spawning season. After spawning, the adults return to the lake from the river, and they leave behind these eggs. The eggs incubate for about a week, depending on water temperature, and then out hatches a free embryo. And as you can see in the middle picture there, the free embryo has a yolk sac attached to it. So the free embryo immediately burrows into the gravelly substrate at the bottom of the river, and it stays there living off of its yolk sac reserves, and then about a week later, it's reached the larval stage, which you can see in that bottom picture. All right, so during the larval stage, sturgeon drift back downstream, going back to the lake. And it's during this time that they encounter a lot of predators. So any stress-related behavioral or physiological phenotypes that they have during this stage that determine how they react to predators, those are going to be important for determining whether or not they survive. So all of my experiments took place at the Black Lake Sturgeon Rearing Facility in Onaway, Michigan. We are studying the sturgeon population that lives in Black Lake, and they spawn in the upper Black River. So every spring, we catch the spawning adults in the river, and we non-invasively extrude the eggs or the sperm, as the case may be, and we bring those back to the hatchery and produce a lot of fish for research. And then we also produce sturgeon to release back into the local population as part of our conservation efforts. So as I mentioned at the beginning, it's important to study stress within relevant ecological contexts. And in my dissertation, I looked at 4 specific contexts. For chapter 1, I looked at the context of the environment under climate change, specifically looking at high water temperature. For chapter 2, I looked at transgenerational context, specifically a match or a mismatch between maternal and offspring environments. For chapter 3, I looked at conservation context, specifically asking whether the hatchery environment itself could potentially be a source of stress. And for chapter 4, I looked at the context of ecological community, specifically interactions with the benthic invertebrates that share spawning habitat with sturgeon. And for all of these, I primarily focused on cortisol and the HPI axis function, behavior, and predation. So moving on to chapter 1, I looked at high temperature. So climate change is one of the major sources of stress for wildlife populations. Great Lakes fishes are facing temperatures that are higher and more rapidly fluctuating. In the Great Lakes region, by the end of the century, there's going to be a 3 to 5 degree increase in temperature, and that's within the lifetime of one individual sturgeon. This is particularly a problem for lake sturgeon because their entire life is very closely tied to temperature. Temperature is what determines their developmental rate during early life stages, and when they're adults, temperature is what cues the beginning of the spawning season. So we already know that lake sturgeon are stressed by warm temperature during early life stages. What we don't know is how that impacts fitness. So I investigated this by taking eggs from sturgeon, fertilizing them, and dividing them between 2 recirculating tank systems. Each tank system had a heater and a chiller to regulate temperature. So the first temperature was cold, and that was kept at 10 degrees Celsius, and the second was warm, and that was kept at 18 degrees Celsius, and that was the high stress temperature. I chose these temperatures because these are the temperature ranges in the upper Black River during the sturgeon spawning season. So these are temperatures that sturgeon could already realistically encounter. And I did that for 4 different families. So I had 6 replicates for each of the 4 families for each of these 2 temperature treatments. And here's the data I collected. At the egg stage, I took cortisol samples. At the free embryo stage, I took cortisol samples and size measurements. And then at the larval stage, I took cortisol samples, size measurements, behavior videos, and I conducted predation trials. And I'll give you more detail about each of those. So for cortisol samples, in order to understand how the HPI axis is functioning, it's important to not just look at the baseline cortisol under normal circumstances where the individual is not stressed, but also look at how the cortisol rises in response to encountering an acute stressor. And looking at both of those reveals how the system is functioning. So to do that, I took 6 larvae from each replicate and euthanized them immediately before they had a chance to react to any stress. And those became the baseline cortisol samples. I also took 6 larvae from each replicate and exposed them to an acute stressor and then waited 30 minutes before euthanizing them. So that allowed the cortisol a chance to rise in response to the stressor. So all of these were stored in these tubes in liquid nitrogen, and then I conducted cortisol analysis using liquid chromatography tandem mass spectrometry with the help of the Wei-Ming Li lab. For size measurements, I quantified size using ImageJ software. For behavior videos, I analyzed behavior using LolaTrack software from Lolago Systems, and I'll give you more detail about that in a moment. And then for predation trials, I took 20 larvae from each replicate and put them in separate tanks. And then into each tank, I put one crayfish. And I left them for 5 hours, and then I counted survivors. So for all of these 4 data sets, I statistically analyzed these using R software to build generalized linear models. I then conducted AIC model selection to identify what were the important predictor variables explaining variation in each data set. I then further investigated the variables present in the AIC selected model using either ANOVA or chi-square. Chi-square was for the predation trials with a Poisson distribution, and ANOVAs were used for everything else. So here's some more detail about the LollyTrack software. So this software will identify each individual in an enclosure, like these Petri dishes, and then it will track the movements of that individual for the entire video. So you can see the green lines in the Petri dishes here. That's the path of movement that that individual is taking. So then LollyTrack gives velocity, acceleration, the percent of time that individual was active, and the total distance that they traveled during the video. So those were the 4 behavior variables that I analyzed. So moving on to the results, here's the full model for size, and here's the AIC selected model for hatch, and for the free embryos, and for the larvae. So you can see that temperature is important in all of these AIC selected models. And then you can see the results of the ANOVA in the p-values on the figure, and temperature does have a significant effect on size at each of the 3 stages. At hatch, the individuals from the warm treatment are smaller, and then that's actually switched by the free embryo and the larval stage. So by the time they get to the larval stage, the individuals from the warm treatment are significantly larger than those from the cold treatment. Then looking at cortisol levels in the fertilized eggs, this is the full model, and it's also the AIC selected model. So you can see that the eggs from the warm temperature treatment had higher cortisol, so the embryos are already producing cortisol before hatch. But the difference between the 2 temperatures is not significant. And then looking at cortisol at the free embryo stage, this is the full model and the AIC selected model. So there is no difference between temperature treatments for baseline cortisol levels. However, there was a significant difference in how individuals responded to the stressor. So you can see the post-stress cortisol levels, there is a significant difference, and the individuals from the warm treatment have significantly lower cortisol in response. So they had a lower reactivity to the stress. And then you'll also notice that both groups experienced a significant increase in cortisol between baseline and post-stress. And that happened for both groups. And that's the cortisol spike that the HPI axis produces in response to the stressor. So this is an indication that the HPI axis is functional. And for the larval stage cortisol, here's the full model and the AIC selected model. So I saw a very similar pattern here where there was a significant cortisol spike for both groups. But at the larval stage, there was no significant difference between treatments. And then looking at behavior, here's the full model for each behavior variable, and then the models for velocity, acceleration, percent activity, and distance. So temperature is important for all of these. And then here's the results of the ANOVAs that I conducted. So I saw that based on the ANOVA results, there was no effect of temperature on velocity or acceleration. However, the larvae from the warm treatment did have a significantly higher activity level, and they moved greater distances than those from the cold treatment. And then finally, looking at predation results, I saw a dramatic difference between the 2 treatments. So here's the full model and the AIC selected model, and temperature is important in that. And you can see the ANOVA results right on the figure there. So you can see that larvae from the warm treatment actually had much higher survival in the presence of a crayfish than larvae from the cold treatment. So here are some important points from chapter 1. I saw that the chronic high temperature stress reduced HPI reactivity, at least during the free embryo stage. And this has actually been seen in other fish species as well. And it's thought that this could be an adaptive response to chronic stress. If you're already experiencing a lot of stress in your environment, then it's useful to limit your response to any additional stressors that you encounter. And that's a way to avoid perpetuating a hyperactive stress axis. And so essentially what we're looking at here could be considered a physiological resilience to stress. And along with that, I saw that chronic high temperature stress resulted in higher activity levels and lower predation rates. So there was a behavioral change, and that behavioral change seems to be adaptive because it's helping larvae avoid crayfish predators. So this is an adaptive behavioral response to stress, at least during the short-term context of predation during the larval stage. So moving on to chapter 2, I investigated the context of transgenerational mismatch. So in fishes, stressed females will provision elevated cortisol levels into their eggs while the eggs are being formed. And the cortisol in the egg does a lot of different things, and it can developmentally change those embryos and set them on trajectories to develop phenotypes that are going to be suited to the high stress conditions. And this is particularly important for species like lake sturgeon that don't provide any parental care, because this is the female's one window of opportunity to set up her offspring to thrive in the environment that she's been experiencing. The problem is, if the environment changes rapidly enough, the offspring environment may not match the maternal environment. So the female may be provisioning eggs in a way that's not actually suited to the environment the offspring are about to find themselves in. And this can be experimentally investigated by combining two techniques. The first is manipulating egg cortisol, and that can be done by incubating eggs in a cortisol solution during the fertilization process. So fish eggs will absorb water from their surroundings during fertilization, and so they will absorb cortisol and end up with this artificially elevated cortisol level. And that's a way to simulate maternal stress. The second technique is manipulating the offspring environment using an unpredictable chronic stress regime. This is something that's been developed for rodent studies, and it's where you have a set of 3 stressors and then you randomly choose 2 of them to apply every day. So the subjects are constantly being stressed, but they don't have a chance to become habituated to the stress because it's different every day. So I applied this during the free embryo stage, and the stressors that I chose were light exposure, a thump on the table surface, and low water levels. So each of these were applied very briefly for 2 minutes or less, but the free embryos do react to them. So I used different combinations of these techniques to set up matches and mismatches, and I ended up with 4 treatments. So for each batch of eggs, I divided them between high egg cortisol and low egg cortisol groups. For the high egg cortisol, I applied the treatment where they're incubated in the solution, and then the low egg cortisol, that group I just left alone. So they had whatever cortisol levels that the female had already put into them, so that was lower. So the high egg cortisol is a match with high offspring stress, and that's the SS treatment that stands for stress. And so for those free embryos, I applied the unpredictable chronic stress regime. High egg cortisol is a mismatch with low offspring stress. That's the SC treatment. And C stands for control there because I did not apply the unpredictable chronic stress regime to those free embryos. Low egg cortisol is a match with low offspring stress, so that's the CC treatment, and a mismatch with high offspring stress, and that's the CS treatment. And I did this for 2 different families, so I ended up having 6 replicates for each of the 2 families for each of the 4 treatments. And with both families, this is the full experimental design. And here's the data I collected. So the egg stage, I collected cortisol samples. At hatch, I measured size and yolk sac area. And at the larval stage, I took size measurements, cortisol samples, and behavior videos. And my statistical methods were the same as in chapter 1, except that I did something different with the behavior dataset. So one thing I did is I used the software to define a center zone in the Petri dish. And this is the entire center of the Petri dish, excluding a 1-inch perimeter around the edge. So in addition to the variables from before, LollyTrack also gave me information about the amount of time each individual spends in the center zone and the number of visits to the center zone. So from each video, I have all these variables, and this is a lot of variables, and they're all kind of related to each other because they're based on the same swimming movements. So instead of analyzing each of these separately, I used a principal components analysis to compress many variables into a few principal components. And then I can see that each principal component is informed by some variables more than others, and I can use that information to characterize each one as some kind of behavior that it's describing. So for example, with principal component 2 in this case, if that's primarily informed by zone time and zone visits, I can characterize that as something like zone avoidance. So then I analyzed each principal component using generalized linear models and ANOVAs to see how those differed among treatments. So moving on to the results, I'm going to go very briefly over the yolk sac and size results. Essentially, there was no effective treatment, but there was an effective family. Here's the yolk sac area results, and it's the same story with size at hatch and at the larval stage. So you can see in the AIC selected models, family is the only variable in those. So essentially, it looks like there are genetic components or other family-specific differences that are driving growth and metabolism more so than these treatments. And here's the cortisol levels in the fertilized eggs. The AIC selected model was the same as the full model. So for these samples, I actually took repeated samples over the first 24 hours after fertilization, and that was to see how the technique had worked. And you can see in panel A, those are the control eggs that were not treated, and then panel B are the ones that I treated in the cortisol solution. So for the control eggs, you can see that cortisol is relatively low the entire time. And then in the eggs that were treated with the solution, there's a large increase in cortisol right after fertilization. So that's a good sign that the technique worked. And then you'll also notice that by 24 hours after fertilization, all of the cortisol has actually been ejected from the egg. So that is actually the normal process in fish eggs. So that is what I was expecting. And I'm still expecting that the elevated cortisol during those first 24 hours is still going to have a developmental effect on the embryos. So here are the cortisol results for the larval stage. And the fully parameterized model was the AIC selected model. So I divided these results between the two families, with family 1 in panel A and family 2 in panel B. So you can see that for both families in all of the treatments, there's a significant increase between baseline and post-stress cortisol. So that's the cortisol spike that's indicating that the HPI axis is functional for all of those fish. But you'll notice that it looks different in this treatment, in the SS treatment. So these individuals had a much lower cortisol spike in response to the stressor, and that is a significant difference compared to all the other treatments. However, that only happened in 1 out of the 2 families. And here are the behavior results from the PCA. The PCA showed several interesting patterns in behavior, but for the sake of time today, I'm just going to focus on PC1, which was primarily informed by percent activity. So here's a result, here's a summary of the results that I saw with that. So the significant difference was between these 2 treatments, the CS treatment that had low egg cortisol, high offspring stress, and the SC treatment that had high egg cortisol, low offspring stress. I saw that fish from the CS treatment had significantly higher activity than those from the SC treatment. So something interesting about that is that these individuals in the CS treatment, they had a very similar situation as the larvae back in chapter 1 with the high temperature, where they experienced a high stress during early life stages. And this is the same behavioral outcome. This is the same increase in activity. But you'll notice that this experiment had another treatment where the offspring also experienced high stress, and that was the SS treatment. And those fish did not increase their activity levels. So potentially there's something about the high egg cortisol that is mediating how offspring are responding to the stress that they experience, and potentially preventing this behavioral change from taking place. So here are some of the important points from chapter 2. I saw that in the treatment where there was high egg cortisol and high offspring stress, that resulted in reduced physiological reactivity, which, as we saw from chapter 1 and based on background literature, that's potentially an adaptive response and can be considered physiological resilience to stress. And that only happened when the high stress in the maternal environment that was simulated matched the high stress in the offspring environment. However, I only saw this in 1 out of the 2 families, so the effects of this treatment were highly family specific. And that indicates that there may be genetic factors or other family differences that are going to be mediating how offspring respond to these different combinations of maternal and offspring experience. I also saw that early life stress increased activity, just like before. but only if the egg cortisol was not elevated. So this confirms that egg cortisol has an important role in determining how offspring are going to be responding to the experiences they have. Overall, the behavior results indicated that it's the combination of maternal and offspring stress that's more important in determining behavior than is maternal stress alone or offspring stress alone. So in other words, the maternal and offspring experiences are interacting to determine what the offspring phenotype ends up being. So in the case where there's rapid environmental changes, then it's likely that this kind of mismatch between generations is going to take place. And we saw today that that can have phenotypic results, and so that is probably going to impact survival and have potentially population-level consequences. And this is a situation that wildlife are going to be increasingly encountering in light of climate change, which causes rapid environmental fluctuations. So looking at the transgenerational context is important when studying stress. So moving on to chapter 3, I looked at the context of conservation programs. So reintroduction programs that release animals into the wild are important tools for wildlife conservation. The problem is that it's fairly well documented that captivity can have a negative effect on fitness. And so individuals that are released into the wild are not going to have very high survival or high reproduction. And that's going to be counterproductive to the goals of the conservation program. Head starting programs like our Sturgeon facility collect individuals from the wild while they're very young and then raise them in captivity and release them later when they're older. The idea is that keeping them in captivity during these early life stages is going to maximize survival by protecting them from threats. But it may also be exposing them to stress during these very vulnerable early life stages. So at Black Lake, we actually have a perfect system set up in which to investigate this. As I mentioned earlier, every spring we catch the adults as they're spawning and we take the gametes back to the hatchery and we produce our own fish in the hatchery. Another thing we do is we set drift nets in the river and we capture the wild sturgeon larvae that were spawned naturally in the wild. And then we bring those back to the hatchery and we raise them in captivity all summer to maximize survival before releasing them in the fall. So side by side in the hatchery, we have these two groups that we can compare. So I compared these fish at 3 times over the first 9 days of the larval stage, day 0, day 5, and day 9, and I labeled these stages A, B, and C. So it's important to note that the wild caught fish that I used for this were only captured from the wild 1 time, and then they were maintained in the hatchery environment over the next 9 days. So I was able to not just compare the hatchery sturgeon and the wild sturgeon, I was also able to see how the hatchery environment influences the wild sturgeon over time as they spend time there. So at each of the 3 stages, I collected data on size measurements, cortisol samples, I took behavior videos, and I conducted predation trials. And I had 6 replicates for each of 4 hatchery families or 4 drift nights, and then in each of the 2 treatments. So I did something slightly different for the predation trials this time. I had one set of trials where I used larvae that were naive to crayfish odor, so they had never experienced it before that I knew of. And then I did a second set of trials that I first conditioned the larvae to a combination of odors. And so one of the odors was made from dead sturgeon, and that contains alarm cues. Alarm cues are substances that are released from sturgeon skin that's injured, and so that serves as a danger signal to other sturgeon. And this is something that a lot of aquatic species have, it's not just sturgeon. So I combined the alarm cue odor from dead sturgeon with the odor of crayfish. And the idea is that lake sturgeon larvae should be able to learn that crayfish are dangerous by experiencing that combination of odors. So those larvae were conditioned with those odors before going into the predation trials. And what I was hoping to observe was not just survival rates, but also how the rearing environment influences their ability to learn to avoid predators. So my statistical methods for cortisol, size, and predation were the same as before. And then for the behavior videos, I used a PCA like I did in chapter 2. And here are the results. So for body size, here's the AIC selected model, which was also the full model. And I saw that for the most part, the hatchery larvae were significantly larger than the wild larvae. And that's something that we were expecting based on observation, and that's probably good because it's been shown before that larger size is beneficial in the wild. And here are the cortisol results. So I saw something really interesting happen here. Here's the AIC selected model for stage A, and you can see that treatment and stress state, baseline or post-stress, are both important for cortisol. And here are the ANOVA results for that. So hatchery larvae actually had lower cortisol than wild larvae. So they may be less stressed. This also may have to do with the fact that the wild fish had just been captured from the wild the night before, which presumably was a stressful experience for them. And you'll also see that there's a significant spike in cortisol between baseline and post-stress for both groups, indicating that the HPI axis is functional. That's for stage A. At stage B, the only variable included in the AIC selected model was stress state. And here are the ANOVA results for stage B. So at this stage, there was no difference in cortisol levels between the two treatments, between hatchery and wild. There's still a significant spike in cortisol, indicating that the HPI axis is functioning for both of these groups. And then looking at stage C, the AIC selected model was actually the null model with no variables. And here are the ANOVA results. So I saw that there was no difference between the two treatments, and there was also no cortisol spike for either of the treatments. So both of these groups actually stopped physiologically responding to stress. And here are the behavior results from the PCA. PC1 was primarily informed by percent activity, PC2 was informed by distance, and PC3 was informed by zone time. And here's a summary of what I saw with those. So hatchery larvae decreased their activity levels over time at each stage, and wild larvae increased their activity levels over time. So they had the opposite direction of change. At all stages, hatchery larvae consistently moved greater distances than the wild larvae. And at all stages, the wild larvae consistently had higher zone avoidance than the hatchery larvae. So the wild fish chose to stay more along the edge of the petri dish. So there were these consistent behavioral differences in not just the ways that they were moving, but in the ways that they chose to use the space. And finally, here are the predation results for this experiment. So you can see in the AIC selected model, stage and treatment are both important. And here are the ANOVA results. So at stages A and B, I saw that hatchery larvae had higher survival than wild larvae. But then by the time they get to stage C, there's no longer any difference between the two treatments. I also saw that for both groups, survival significantly decreased at each stage. So more of them are being eaten by the predators over time, until stage C, when they both have similarly low survival rates. So here are some of the important points from chapter 3. I saw that captivity can have an effect on wildlife over a very short period of time. It only took 9 days for the wild-caught fish to resemble the hatchery-produced fish, both in terms of physiology and in terms of their survival. And this is important because a lot of the literature looking at the negative effects of captivity look at these effects over a very long period of time. And one example is domestication selection, which is a large concern in captive wildlife, and that's something that happens over multiple generations, so a long-term span. And this study showed that this happens for individuals within only a few days. I also saw that there were consistent behavioral differences that did not go away. And this suggests that there's important influences on behavior that are happening prior to the larval stage. So it's probably things during the egg stage and the free embryo stage that are programming these behaviors. It's also important to note that cortisol levels did not indicate that the hatchery causes chronic stress. So that's good news for us. But captivity does appear to decrease predator survival. And I also saw that neither group was able to learn to avoid predators based on the combination of odors. So this implies that conservation programs should certainly consider animal welfare for their captive wildlife, but also go beyond that and find ways to promote behaviors that are going to be adaptive after those individuals are released into the wild. So in other words, just because an animal is happy doesn't necessarily mean it's prepared for the wild. So moving on to chapter 4, I looked at the context of ecological community, specifically looking at interactions with aquatic insects. So interspecies interactions can be important influences during early life stages, especially if this involves predation risk, which can potentially be a source of stress. Lake sturgeons spend their free embryo stage burrowed into the gravelly substrate at the bottom of the stream, and this is a habitat that they obligately share with a huge variety of aquatic insects, some of whom are harmless and some of whom are predators. So for example, you can see in the top picture, that's an isonychiid, and that's a filter feeder. And then in the bottom picture, that's a purlid, and that's a predator. So I set up an experiment where I exposed free embryos to an insect treatment for 3 days. And the control treatment had no insect. And then in the isonychiid treatment, I had 6 replicate tanks, each containing 25 free embryos, and then into each one I put 4 isonychiids. So the sturgeon and the aquatic insects were allowed to just freely interact for those 3 days. And then in the purlid treatment, I had the same setup with 4 purlids per tank. So I had 6 replicates for each of these 3 treatments, and these all represented one family. So after 3 days, the free embryos had matured to the larval stage. And at the larval stage, I looked at mortality, size measurements, cortisol samples, and behavior videos. So for behavior videos for this experiment, I videoed 6 larvae from each replicate. I saw their swimming activity in the Petri dish without any extra stimulus applied. And then after 4 minutes, I added an odor to the center, and this is the odor that's made from dead sturgeon containing alarm cues. And then I took another video for another 4 minutes. So I wanted to compare not just their swimming activity with no extra stimulus involved, and then how they behaviorally react to this potential danger signal. So I used the same statistical methods as before for cortisol dataset and the size dataset. And then I used a PCA the same as before for the behavior videos. And looking at the mortality based on insect predation, I used a beta regression to compare proportions, and I did pairwise comparisons to see differences among treatments. So here are the mortality results. I saw that free embryos in the purlid treatment had significantly higher mortality than those from the control treatment. So this confirms that purlids are sturgeon predators and they eat a lot of them. I also noticed that in the isonychiid treatment, some of the free embryos had gone missing under mysterious circumstances. And I realized that isonychiids are actually facultative predators. So some of those isonychiids decided to switch from being filter feeders to preying on sturgeon. Looking at body size, I saw that individuals from the purlid treatment were significantly larger than those from the other 2 treatments. And this is probably because the purlids were preying on the smaller ones. And then looking at cortisol results, I saw this beautifully clear pattern where the amount of stress seems to be proportional to the amount of predation that was experienced. So you can see in the control treatment, those individuals had fairly low cortisol at baseline, and they also had a fairly low cortisol spike in response to a stressor. In the isonychiid treatment, those individuals had higher baseline cortisol and higher reactivity in response to a stressor. And then in the purlid treatment, those sturgeon had the highest cortisol and the highest reactivity to stress. And here are the behavior results from the PCA. So I ended up dividing the behavior data set into 2 data sets. And the first one was looking at the behavior before any extra stimulus was applied. So that first 4-minute video. And then the second data set was after the alarm cues were added. So there were 2 different PCAs. In the first PCA, PC1 was primarily informed by percent activity. And in the second analysis, PC1 was informed by zone visits. So for both of these, I saw that the fish that had encountered isonychiids actually stood out from the other 2 treatments. So to start off with, they were significantly less active than those from the other treatments. And then after the alarm cues were added, the larvae that had encountered isonychiids avoided the center zone more. So they were choosing to stay more along the edge of the Petri dish. This probably has to do with the fact that the alarm cue odor was added to the center of the Petri dish. So those sturgeon that had interacted with isonychiids were more reactive to that danger signal and more interested in avoiding it. So here are some of the important points from chapter 4. I saw that the stress levels appear to be proportional to the amount of predation on nearby conspecifics. And this makes intuitive sense. If individuals are able to perceive a high predation risk, they respond by becoming stressed. I also saw that the sturgeon that had interacted with isonychiids had more of a behavioral change, despite having fairly low mortality in that treatment. And they had that lower activity, and they were more reactive to the alarm cues. So I'm not sure exactly how to explain this, but one possibility is that encounter rate might be important. So the sturgeon that encountered isonychiids frequently were not actually that likely to be predated because the mortality rate was still really low in that treatment. So by the time that they were used in the behavior trials, they had that experience of frequent encounters with an aquatic insect. And then on the other hand, the sturgeon that had encountered purlids frequently were pretty likely to be eaten by the purlids and not survive to be used in the behavior trials. So that may explain the behavior results that I saw. So aquatic insects are really interesting because they're extremely responsive to changes in the environment. So things like water quality, habitat disturbance, and temperature, those are going to impact aquatic insect abundance, biodiversity, and behavior. And all of those, as we saw, are going to change lake sturgeon survival and lake sturgeon phenotypes. So essentially, lake sturgeon are experiencing direct effects of environmental factors, but they also are going to experience indirect effects of changes in the environment through interactions with these other species. And of course, the other side of the equation that I didn't investigate in this experiment was what informs the predator behavior. So for example, I'm super curious to know what causes isonychiids to switch from being filter feeders to being predators. I don't know what it is. I think this is a really fascinating, complex system in which to ask questions. So that concludes my 4 chapters, and I just want to end today by highlighting one of the interesting takeaways from each chapter. And I also have a few future directions to mention as well. So in chapter 1, we saw that early life stress can be useful in promoting adaptation that helps individuals survive future threats. In chapter 2, we saw that maternal and offspring experiences interact to determine offspring phenotypes. In chapter 3, the findings implied that conservation programs should consider not just the stress level of their captive wildlife, but also find ways to promote adaptive behaviors that will enable those individuals to survive after release. And then in chapter 4, we saw that interspecies interactions are important influences on phenotypes and survival. So all of these point to the importance of the early life stages in determining fitness, because it's the experiences that happen during these stages that are going to induce different phenotypes, and those phenotypes are going to make differences in survival. And as we saw today, stress is a really important mechanism for how that happens. And then as always in science, all of these conclusions just bring up yet more interesting questions. So chapter 1 brought up the question of what are the long-term costs to these short-term adaptations to stress? Chapter 2 brought up the question of what is it that mediates the family differences in transgenerational stress? Chapter 4 brought up the question of what is it in the rearing environment that promotes or inhibits anti-predator learning, especially if it's not related to stress. And chapter 4 brought up the question of what are the environmental impacts informing predator behavior that are going to be driving these predator-prey interactions. And all of these questions, of course, point back to the importance of investigating stress within relevant ecological contexts, like we mentioned at the beginning. Stress can be studied in a lab setting with rodent models, and that's a great way to understand things like the physiological mechanisms involved. But to really have insight into whether the outcomes of stress are adaptive or maladaptive, and really understand the consequences of stress, we need to ask questions within a larger context. So species like lake sturgeon that are threatened wildlife are actually really valuable and important species with which to ask these questions and to get a better understanding of the role of stress within these very complex ecological systems.